we are discussing about that if any company manufacture any tablet, it may be ciprofloxacin tablet, it may be metformin tablet, it may be paracetamol tablet. If they want to send this tablet into the market, we need to check the quality of these tablets. That quality test parameters is classified into two types, right? One is wet analysis and second one is chemical analysis. That means any tablet, it may be metformin or ciprofloxacin or paracetamol. Okay, so after completion of the any product in the pharmaceutical industry, if you want to send into the market, we need to check the quality, right? That quality test is classified into two types. One is wet analysis, second one is chemical analysis. How many tests do we have in the wet analysis? 7 tests. How many tests do we have in the chemical analysis? 5 tests. That means totally how many tests we have? 12 test parameters. That means after manufacturing of the product, these quality control people perform these 12 quality test parameters. That means 7 are wet analysis and 5 are chemical analysis. All the test parameters or results are within the acceptance criteria. That means within the limits. Then we send the product, we give the acceptance criteria, acceptance of that product, then the product goes into the market. So you people are already attend the wet analysis classes and you people are already did the wet test practicals also. So you are here to perform these chemical test parameters. So what are those? First one is essay, second one is pronouns, dissolution, UOD, impurities, residual solvents. So totally you have 5 chemical tests. One is assay, dissolution test, uniformity of dosage test, impurity test and residual solvents test. First I explain clearly what is assay, what is dissolution, what is UOD, what is impurity, what is residual solvents. So tell me what is assay? To find out how much percentage of the drug present in sample. అంటే మన టాబ్లెట్ మనం వేసుకునే టాబ్లెట్ లో డ్రగ్ ఎంత ఉంది అని చెక్ చేసే టెస్టే ఎస్ఏ. దెన్ వాట్ ఈస్ రిజల్యూషన్? ఎస్, how much percentage of the drug released in a particular time in a particular solution is nothing but a dissolution. That means if you take a tablet, the tablet goes into the target of release, maybe stomach, maybe small intestinal. The first step was the tablet is going to disintegrate into the small fragments, then the drug is going to absorb into the blood circulations. That process we call as a absorption. In the dissolution we are finding that how much percentage of the drug enter into blood circulations. And then what is UOD? Uniformity of doses. That means all the tablet is contain the equal amount of the drug or not. See, if you are taking ciprofloxacin 500 mg, let us assume that. That means each and every tablet should contain the 500 mg. Yes or no? One tablet should not contain the high amount, one tablet should not contain the low amount. In the UOD, uniformity of dosage. That means all the tablets should contain the uniformity of the drug is there or not there. That's the reason we are performing the UOD. And then what is impurities? To find out how much percentage of the impurity present in sample. That means in the tablet, other than the drug, other than the excipients, if there are any foreign materials are there, that we are finding in the impurity. What is residual solvents? To find out how much ppm of the solvent present in sample. Ante mana tablet lo ఏమన్నా సాల్వెంట్స్ ఉన్నాయా, ఉంటే ఎంత ఉంది అని చెక్ చేసే టెస్టే రెసిడ్యువల్ సాల్వెంట్స్. అంటే ఎస్ఏ అంటే ఏంటంటే టాబ్లెట్స్ లో డ్రగ్ ఎంత ఉంది, ఎస్ఆనో. డిజల్యూషన్ అంటే ఏంటంటే టాబ్లెట్ వేసుకున్న తర్వాత ఎంతైతే డ్రగ్ ఉందో అంత డ్రగ్ బ్లడ్ లోకి ఎంటర్ అవుతుందా లేదా. యూఓడి అంటే ఏంటంటే అన్ని టాబ్లెట్ లో డ్రగ్ సేమ్ ఉందా లేదా. ఇంప్యూరిటీ అంటే ఏంటంటే టాబ్లెట్ లో డ్రగ్ కాకుండా ఎక్సిపెన్స్ కాకుండా ఇంకేమైనా ఫారెన్ మెటీరియల్స్ ఉన్నాయా, ఒకవేళ ఉంటే ఎంత పర్సెంటేజ్ ఉంది. అండ్ దెన్ రెసిడ్యువల్ సాల్వెంట్స్ అంటే మన టాబ్లెట్లు ఏమైనా సాల్వెంట్స్ ఏమైనా ఉన్నాయా అంటే ఎస్టోన్ కానీ టోలిన్ కానీ, ఇఫ్ దెర్ ఇస్ ఎనీ సాల్వెంట్స్, దట్స్ వి ఆర్ ఎస్టాబ్లిషింగ్ టు ద రెసిడ్యువల్ సాల్వెంట్స్. సో that I explained very clearly. Okay, after that I discussed something related to that. If you want to perform these all chemical tests, you require the instrument. Yes or no? Without instrument we cannot perform these tests, right? So out of assay test and dissolution test and UOD test and impurity test, so which instrument we are using? HPLC, high performance liquid chromatography. Yes or no? And final test, this only residual solvents we perform by GC, gas chromatography. That means if you want to perform these 5 chemical tests in any quality control department, so out of 5 testers, 4 testers we perform by HPLC, high performance liquid chromatography, only 1 test we perform the gas chromatography.